we are talking here about mutational resistance. Resistance in general can be a vertical gene transfer or a horizontal gene transfer. In case of these genetic events and antibiotic selection pressure, we have usually we are moving from wild type cells that are susceptible to resistant populations that may be less fit than the original cells, but if more selection pressure is there, then we can find compensatory mutations, and the population becomes much more fit, and in the end, we have resistant cells that are fit. And that's the evolution of resistance and fitness that we can see. Here, in drug discovery, I'm focusing now on the resistance emergence through mutation. And this is the field that's usually explored in antibiotic R&D. What's really interesting for all of us is can we predict the clinical effect? That's what counts, and this is a really complex matter. So first, just to explain what we are talking about, frequency of mutation means the proportion of mutant bacteria that are present in the culture. And what we usually see is frequencies that are below 10 to minus 6. This frequency can fluctuate during the growth of the population, and depending if the mutation occurs very early in the growth or later on, we have different mutation. We find different mutation in this population. The mutation rate is an estimation of the probability of a mutation occurring per cell division. So that's different to frequency of mutation. This mutation rate is usually included in the frequency of mutation. Fitness means that bacteria are very adaptable to the environment, and we see this phenomenon of compensatory mutations. And the bad news is that they may be even more fit than their susceptible revertants. Now, this example that you may know, this is the drug, the boron drug that was discovered by Anacor and then in-licensed by JSK with an in vitro frequency of spontaneous single-step resistance with about 10 to minus 7. These mutants were seen in a large variety, all in the target, and they were high-level resistance. No fitness cost. This drug went into phase 2 for UTI, and resistance developed very rapidly in almost a quarter of patients, with a very high increase in MICs. So that this was really kind of surprising, because on the second day you could already see the rise of MIC from 0.25 to 512. So a really big event. And in the clinical situation, so what was leading to this clinical resistance? It was a high-level resistance with no obvious fitness cost. We have a high burden of bacterial cells in UTIs, so that's an important factor. so that the indication that you are studying, and the genetic plasticity of the target gene. It's not clear if the bacteriostatic nature of this drug has an implication for this clinical resistance. And a similar case, it's less documented, and we don't have enough information. The mutations were very species dependent, also in the target. They were highly resistant, but less fit. But compensatory mutations occurred within 30 to 60 generations of growth, and they were fit in animal models. So this may all have contributed to stopping this clinical program. An example now for fosfomycin, that's an old drug and very well characterized, and we are using it usually for severe infections only in combinations, but this here is the example of the oral version, and it was studied in a dynamic bladder infection in vitro model that simulated pharmacokinetics in humans. And you can see that before and after, so the second measurement for E. coli, the MICs didn't change too much, but they changed a lot in some strains for Klebsiella and also Enterobacter. So also here, it's strain specific and also species specific. And this can be illustrated here, also fosfomycin in this mutation frequency study with several strains, and you can see this variety. So that's the reason why there is no one number of frequency of mutation, but you have usually quite a range of results. And also in Klebsiella, in this case, much worse than for other species. But the message is apparently you need to study a lot of strains to get a picture of what's happening. So finally, how do we predict clinical resistance? And I have here these two examples, which are both single target drugs. Mesillinam, a very old penicillin, and this boron drug from GSK. The frequency of mutation is not good in both cases. They have large mutational targets, so a lot of genes can be involved in mutations, and they were both studied in UTIs. And mesillinam is still used for the treatment of UTIs. So why does mesillinam work clinically and the GSK compound doesn't? And both are used for UTIs. Both are single target drugs. So in case of mesillinam, the laboratory mutants did not predict the mutants in clinical strains. So the clinical mutant was not target-based, and that's a different to the GSK compound. The laboratory mutants predicted the mutants in clinical strains, and there were many different places to mutate, and they were all in clinical strains. In mesillinam, all mutants were fit, but the the compensatory mutations were associated with a loss of resistance in the majority of cases. So they were compensatory mutants, but they were not good in the clinical situation. In contrast, the mutations here supported the maintenance in the urinary tract. And these mutants were not resistant in urine. So a big difference between the lab situation and the clinical situation. This here were no fitness cost and stable. So this is just an illustration how you could make not the best predictions if you have just very little information here, because both would look very similar. So you need to go into much more detail, and stopping here would not predict the clinical situation. So with this was just a very short introduction into this topic. And we will hear now from Michael Mourez about good models and what he would recommend to do and how to study the risk of mutational resistance.
Why do we need actually to look at resistance in a broader sense? It's because I think there's a renewed interest in phenotypic screening, and so when you do those, then you end up with compounds where you don't know the target. That's a fact. Secondly, the interest in gram-negative bacteria also makes new approaches where you want to break that barrier by using Trojan horse approaches and things like that. So antibiotic hybrids, you complexify a lot the mode of action, and you have to take into account the way the compound penetrates as a means also for resistance to emerge. So that's why I think it's very important to have those tools where you look at the resistance potential in a very broad sense, rather than looking just at the target when you actually know the target. I will go very fast and try to show you, I'll give you examples on how to assess resistance, then understand resistance, then translate resistance, and then manage resistance. First, if you want to assess resistance, well, why do you do it? Well, first, because you have to. Right? The authorities, and I'm not speaking for the authorities, but this is something coming from EMA, and the guidelines that have been published in December, they tell you that you have to look for resistance. So the first thing you should do, or you can do, is to do just MIC90s and on a broad panel of strains. And if you go one step further, it would be very interesting to see if the MIC of your compounds actually correlate with the MIC of known compounds, hinting at the ability or the potential for cross-resistance, as shown here. So basically, if you've got a compound where with a very narrow distribution, it looks pretty good. If it's very spread out, then you may have a problem, or at least you need to understand what's going on. And similarly, if you have a compound whose the MIC of which correlates very well with ciprofloxacin, chances are that you're targeting gyrase. So here you might have an issue. Then we go to the workhorse of the resistance studies, something that we all do, going at this frequency of resistance, of spontaneous resistance. The experiment goes like this. You plate as many bacteria as you can, and there is lots of arguments how many bacteria should you plate. And you do that on one strain, and you look after a period of time, again, 24, 48 hours, depends what your religion is, and you count the number of clones. And from that, you derive a frequency of resistance. First thing is that you should be wary and check always the stability of the clones you count. Something that many people actually forget to do because they want to go fast. But you should pick the clones, put them on a non-selective medium for at least 2-3 times, and then reprobe whether they're resistant or not. As illustrated here, you see that the clones that are picked for rifampicin, for instance, in different strains are all very stable. For kanamycin, it's really not the case. Basically, the kanamycin resistance doesn't stick when you're not putting pressure. And for nalidixic acid, it's very variable. So be very wary about this problem of stability of the resistance, because it confounds the results. Then all the clones that you have should be characterized in terms of MIC shifts, cross-resistance, and fitness, as said by Ursula previously. There are alternative ways to generate mutants, and because, as said also by Ursula, if the event that you're looking for is appearing early in the preculture, you will have a lot of resistance. And if it comes late, then you will have less. So there's this fluctuation, and so an alternative is to do this fluctuation test, where the output is not only a frequency of resistance, but also this mutation rate using a mathematical model that was proposed in 1943 by Delbrück and Luria. Another way, and we tend to use those serial passage experiments. So the idea here is just to take the bacteria, submit them to pressure, and then at half the MIC, then regrow them and see if the MIC can increase after days of passage. This is something usually that's a bit more difficult to understand. It's very hard to translate that into a frequency of resistance. The only thing you have is a makeshift at a passage number. And so what do you make of that? A bit more complex. And you've got multiple mutations at the same time. So it's a bit harder to interpret. But when you don't have a frequency of resistance, or when your frequency of resistance is below 10⁻⁻⁻, whatever, usually going to the passage experiments is a pretty good idea because you do want to know whether you can get resistance or not. There are new ways, and you maybe have seen this very cool and nice system devised by Roy Kishoni, which is a swim plate, a very big swim plate, where you've got increasing concentration of an antibiotic. Here it's trimethoprim, and you have a camera that looks at the time it takes for the bacteria when they hit the next gradient to spread resistance and go further and further. So here again, you've got a number, the time to MIC shift, but what do you make of that number? Very complicated also to make sense of it. It's nice, but what does it mean? A bit harder. Switching gear, now you've seen how you can actually generate the mutant, look at them and get numbers and how frequently they appear. So now the next challenge is to understand why they came in the first place. And here again, why would you want to do that? Because you're told to. If you look at, the EMA guidelines, mechanisms of resistance should be investigated, so you have to do that. So if I go back to the MIC 90s, if you've got a clinical strain that have higher MIC, you should try to understand why, and that is extremely complex. Because if you do genome sequencing of that, which is something we routinely do, there are so many variants, of course, in a clinical strain genome that it's very hard to understand what in that genome actually makes for resistance in a new drug, in a known drug. It's already hard enough. I'm not even speaking for bioMérieux colleagues who could certainly say, hi, Alex, who can tell you that it's actually hard to take a genome and derive an MIC for a known compound. If you have a completely new compound, it's even harder. In some cases, we've managed to do things like we had a compound that have different levels of activity, again, a panel of strain, and looking at some, we knew a little bit the targets. So looking at the targets, target-rated gene, we could derive a tree where you could clearly see there is a differentiation between the non-responsive and responsive strain, whereas the general tree is not separated like that. So this is by no way a proof, but it tells you, it suggests that this gene is actually should be involved in resistance. So it's very, very difficult to make sense of this data. Of course, sequencing the resistant mutants you've generated actually usually gives you more information. And this is an example where sequencing a lot of mutants, you see, and this refers to what Ursula also was saying before, we did see mutants in Gen X, but we also saw these other events that you were alluding to, that insertion sequences getting in front of Gen X and making the expression of Gen X much higher. We saw also duplication and sometimes to a very large degree of the Gen X itself. So even when you do a very raw, spontaneous frequency of resistance experiment, you can see also these kind of weird events, not only mutation, but also this amplification of the gene. And sure enough, Gen X is the target. You can have a much more focused approach also, because most of their compounds, all of our compounds, are actually subject to efflux and influx issues. And so, of course, you could also just look at all the pumps and then delete them 1 by 1, 2 by 2, 3 by 3, whatever your fancy is, and then look at the impact that you have on different compounds. You could also weaken the outer membrane, which is also something you can routinely do. You have different genes that you can knock out and then make the bacteria more permeable, and you see what is the impact here on your MIC. For instance, in that compound here, the last compound, the compound 3, you see that efflux doesn't seem to be an issue, but here when you weaken the membrane, you have got much more influx. So probably here a rate-limiting step for your compound is influx. We have also a new tool, well, relatively new tool, which is transposon sequencing. And I'm not going to describe the principle of transposon sequencing. It's illustrated here. But the point is that you can actually look at the whole genome of a bacterium and test in one go what the impact of each gene can be on the MIC of your compound. Looks fancy, looks very easy. It's actually a little bit more tricky than it looks like. But this is what it can do to you, for instance. Here, and I invite you to come to the poster I will present during the days to have more details. But here, this is a measurement of the abundance of the transposon mutants with or without treatment with the compound at its MIC. And you see mutants that have enhanced fitness and mutants that have reduced fitness. And what's interesting here is that you can see that all the, well, you cannot read, but you see SCRB, SCRA, TOL-C, so the efflux pumps are all there. So when they are knocked out, the mutants are actually reduced fitness. So this tells you that your compounds probably efflux quite well, because this is spectacular, going out spectacularly well. And sure enough, you can see that when you do the MIC with a TOL-C mutant. But here, when you look at the right hand, there is one gene here that caught our eye, which is called SBMA, and its sensitivity to B17 microcin, so it's an inner membrane transport system. And so this suggested right away that this is probably the influx mechanism of the compound. And sure enough, when you knock out that transporter, you completely blow out of proportion the MIC. There is many other genes, of course, to look at, and it then becomes harder to make sense of them. In many cases we can, but it's more speculation. What's interesting is that you can use that also as a starting point to optimize your molecule. And so this is looking at the actual transposon and how, from an untreated case, when you treat with the native molecule, you see that all these genes insertions in SBMA are favored. And conversely, in TOL-C, when you take them out, when you have the native molecule, they are clearly taken out by the treatment. If you optimize the molecule and you do the test again, you see that this molecule, for instance, is much less dependent on this inner membrane transport, and it becomes less susceptible to efflux. How did that trick was accomplished? Very easily, by putting in a siderophore, which you can clearly see here, because then the strain the compound becomes dependent on siderophore receptors in the outer membrane. So you see that the transposon sequencing system actually allows you to, in one go, to look at many things at the same time. Okay, switching gear again, and I again apologize for going very fast, but how do you translate that from in vitro to in vivo? And why do you want to do that? Here, you don't have EMA guideline, but it just stands to reason. It, again, echoes what Ursula was saying before. Try to make sense of what you gathered as in terms of information, and test the in vivo fitness, for instance, in order to see if the in vitro knowledge that you've gained can actually transfer to the in vivo situation. We also start to do experiments where we want to select and observe resistance directly in animal models. And this is actually very tricky also. So things we can do and things we do, for instance, is to compare in vitro models to in vivo models. And we can do that using, here we use RNA-seq studies, for instance, to compare the different systems. Because then, if we know our target is or the genes that are involved in our compound are somewhere in this heat map, we can see if the same genes are important in vivo also. And we have then a rationale to say, is that going to happen in vivo or not? We also use a biolog system, Omnilog system, which allows us to probe the activity of a compound in 1,000 different conditions at the same time. And here we can then gain some information, for instance, comparing treated and untreated bacteria at different conditions. And sometimes we see that in some conditions, the compound are actually much more potent, for instance. And that tells you something that maybe this condition actually alters the activity of your compound, and that can give you some clues. For instance, in that case, we see that urea was really potentializing a lot the activity of the compound. So maybe the compound was better active in urine, and surely it did. As I said, we also want to look at mutants that could be selected in vivo directly. The issue there is that usually you challenge your animals with a low inoculum size, because otherwise you kill them too fast. And so usually it's 10 to the 6 or 10 to the 7 CFU per animal. So how do you go as high as 10 to the 9 or 10 to the 10 CFU, which is the amount of bacteria you use in the in vitro systems? So there are different ways that we are investigating, and I will not say much more, but in some cases, you don't even need to go that high. And so, but usually in the thigh, you can actually inoculate quite high. You can go to 10 to the 8, and the animals survive in that case. And in those cases, for instance, you can actually see that in some cases, when double plating, you can see that resistance are actually present in the population. And lastly, I will allude to the fact that in vivo, of course, compared to the in vitro systems, you have varying concentration of the compound. And so this is something that we advocate for usually late in the game, but it makes a lot of sense to see what happens when you actually change the MIC of the compound to mimic the in vivo conditions. Because it is also something like an hypothesis that we all make, and it makes a lot of sense that the compound at sub-MIC will not impact the frequency of resistance. And this is also something that sometimes, in some cases, might not hold true, especially when your compound induces SOS response, for instance, and then changes dramatically the frequency of resistance or the mutation rate. Last point. And this is something we usually also advocate for late in the game when you're reaching clinical candidate stage, how to manage resistance. Now you've garnered a lot of information, what can you do? Well, the first thing that we advocate for is to look for a dose. Okay, so let's say you have a bad frequency of resistance, and does that kill your program? Well, not necessarily, because in some cases you can find a dose that prevents resistance. And there are different ways of doing that. The easiest way is that you target for resistance frequency, here 10 to the minus 9, and then you play it at 4 times the MIC, 8 times the MIC, 16 times the MIC, until you don't see anything that grows. In that case, 32 times the MIC. So basically, you know that if you dose high enough, then you don't have mutants that come up. Something you can also, and we think it's better to do that as a time kill curve, because here you have more vision of what's happening really in the population. And similar thing here, 32 times the MIC, you have nothing, no mutants that grow. You can also try to manage the resistance if you have the mutants in vivo. You can actually also look at how you can manage that by changing the dosing regimen in the animal. And this is dose fractionation studies where the compounds are given at a dose once or 6 times a day. And each color is the same total dose per day. You see that's the total CFU, and those are the resistance CFU. So again, it's always a double plating. Here you kind of clearly see that when you fractionate the dose at intermediary dosages, you actually favor resistance. So this is something, I mean, it's interesting to know because it doesn't maybe strike you as much when you look at the total CFU, but here you know that it's something you should avoid. So clearly, doing the double plating in vivo can actually tell you stuff. And of course, here, the hollow fiber is very, very important, although sometimes it just brings a lot of question. Like, for instance, here it's results from a hollow fiber study in tuberculosis, in M. tuberculosis, where for rifampicin, where AUC over MIC drives the activity. And then when you look at the resistance appearance, Cmax over MIC drives the resistance. So there, you're like, what do I want to optimize first? And then the last resort is really to look at combinations. So basically, you've got your compound, you cannot manage the resistance, so should you look into compounds that could prevent resistance from appearing? This is a paper from Roy Kishoni that promotes this idea. Honestly, it's a bit difficult to implement, but maybe later. Maybe when we are tired of throwing away compounds because they have poor frequency of resistance, will we all as a community say, maybe it's time to start doing like what's done in TB and just do combination, but maybe in the future. So I will leave it at that. Again, sorry for being very fast. As discussion point, I think I alluded to them as I was talking. There are a number of points that you need to take into account. All of these experiments that I've briefly exposed to you have variability, either experimental or genetic. For instance, here, this is illustrated here, the genetic background of E. coli, whether you have a wild-type strain or ACRB mutant, that has an impact on the frequency of resistance that you see in spontaneous experiments, as quantified here. And there are many different genetic contexts that we know impact the frequency of resistance. So there's a lot of variability. So then it begs the question of the sampling. You know, how many strains? What are the different strains that you will pick in order to have a better view? And sometimes the differences can be very, very significant, like the examples with fosfomycin that Ursula presented to you, where you have 8 logs almost, or 6 logs differences between strains. There are many, many options, and again, time and cost are things you have to bear in mind. And all of that for a partial answer, because we're only looking at what's happening in the test tube or in an animal model where with isolated strain. You don't look at things such as much more difficult, like persistence, tolerance, and horizontal evolution also risk. There are some protocols, one shown here, where you can try to do that. And here the protocol is just to take environmental sample, shear the DNA, and then force the DNA to be kind of expressed or present, and then transform the bacterium and see if you have a piece of external DNA that will increase the MIC. This is a very long experiment, and how do you interpret that? Very complicated. So the last point is actually, I think, the most important one to take home with you is, how do you translate all of that? As Ursula was saying, you have to take a very holistic view, and there is no, to my knowledge, consensus. Because intuition, for instance, if you take only 2 parameters shown here, where it's a study where each circle represents a resistant mutation and its size, its rate of isolation, you have whether the impact is very high or minimal in terms of MIC, and whereas whether the resistance level is going to appear fast or not. Intuitively, if you go up there, then you're in a very bad spot. But sometimes intuition really doesn't translate to the clinical situation, as Ursula already said to you, and this is shown in this slide.
What do we mean by single target antibiotic? We mean one drug, one gene, one product. Okay, simple as that. Classic example, rifampicin. One gene, one product, one drug. What about resistance? Well, there's different sources. It's been mentioned many times, spontaneous mutations, and that's the focus, and that's what EMA, as we've been told, demands that we measure. But there's also pre-existing resistance in a population that could be selectively enriched, and then there's also horizontal transfer from bugs you're not necessarily trying to target that might eventually affect your drug. Complications. Some of the TPP pathogens that you're targeting may have more than one gene for the product. So even though it's a single product, there could be multiple genes. Drugs that you think are multi-targeting may be effectively single-targeting if you have unbiased coverage. And finally, and this has been mentioned, resistance very frequently occurs off-target by efflux, influx, horizontal transfer, etc. So, wrong one. Background question, so then, are single-target drugs unacceptable in terms of probability of mutational resistance? There's a biological risk here, an economic risk, because many of you are people trying to develop drugs. And there's also medical risk, and I think this was possibly one of the reasons why GSK pulled that program, a combination of economic and medical risk. If you're going to have failure, it's not good for your reputation. The big question, can you predict resistance, and what studies should you use? Well, Michael has gone over many of these. Frequency of resistance. You can have single selections at different concentrations. You can have multiple ascending selections. You can do serial passages. Again, you can vary this. You vary the concentration. You can have ascending concentrations. You can vary the population size. All of this is going to influence the outcome of the experiment. And this goes back to what you said. It can be very difficult to interpret these things. You can search for resistance and find it, but what's the take-home message you should take? Similarly for hollow fiber here, complex system, useful probably later in drug development, but most of you are going to be concerned about very early stage, should I continue with this compound or not, or is this just going to be a dead loss? What can you do to mitigate the resistance risk? Well, of course, you measure frequency resistance. You can do MIC90 measurements to see what's in the population. You can check fitness. You can look for compensatory evolution if you want to go that far. And I'm going to come back to these things because you can really ask the question, you know, does fitness matter? Does compensatory evolution really matter outside of the lab? And then why does any of this matter, besides EMA asking that you do these measurements? Well, it could affect your choice of pathogen, definitely will affect the economics of development if you run into problems later on. And then underneath all of this is the issue of you're measuring mutational resistance, but horizontal transfer is effectively not being measured at all, except in indirectly in MIC90 measurements. Wrong one again. Okay, so let's just consider mutations versus horizontal transfer for a start. If you look at acquired genes, in Enterobacteriaceae, that's overwhelmingly the way we have resistance with the beta-lactams. So the most important class of antibiotics we have, it's got effectively nothing to do with mutational resistance, except that you have mutational evolution once the resistance genes have been acquired. For the quinolones, you have horizontal transfer also affecting it, but also mutations. And for bugs like Streptococci and Neisseria, when it comes to beta-lactams, it's horizontal transfer, but it's in a mutational sense. It's not acquiring plasmids. And then classic mutations, MTB, the most important example where it's only mutations, but mutations in the Enterobacteriaceae and in the gram-positive strep and Neisseria. So both of these are important, and I think it really is going to depend on the drug and the bug. And the reason I bring this up is at an early stage, you still don't really know what you're going to be doing with this compound, where it's going to end up. And you're going to have to decide, is mutational resistance going to be the big issue or not? What are the effects it has if we think of single target versus multi-target drugs? Well, I've put up a list of causes of resistance, and target mutation rate, yes, that will definitely be lower if you have a multi-target drug, just first principles. But pretty much everything else, it's not going to make much difference. Efflux doesn't care, influx doesn't care, drug modification enzymes don't care. And to a very large extent, target protection mechanisms will be indifferent to whether or not your drug is multi-target or single-target. So when we focus on spontaneous mutation rates because I think not only we're asked to do it, but that's what we actually can do. The other things are so much more difficult to measure and evaluate. Keep doing the wrong button here. Okay. It's inevitable that mutational resistance will occur. And I put up these numbers. Ursula mentioned one of them earlier. But spontaneous mutation rates, any nucleotide anywhere in pretty much any genome is going to change at about 10 to the minus 10. Some examples up there. When it comes to inactivating mutations, so things that regulate efflux pumps, I should have put in outer membrane porins as well, where a simple inactivation will do the trick. We're looking at 10 to the minus 7, and that's going to affect most drugs that come into development. The issue is, will it have a fitness effect as well that might counter-select it clinically? And then duplications, things like beta-lactamase genes, efflux pumps, these are extremely frequent. And again, almost any bacteria, anywhere that you're going to target, these things will happen in frequencies which are in that 10 to the minus 2 to 10 to the minus 5 range. They'll be unstable. They may not cause a huge increase in MIC, but they're underlying all the time and they're just continuously occurring. Same one again. Now, the actual rate per gene can be much higher than the mutation rate because most genes will have multiple places where a mutation can affect the drug. And there's one additional thing. Out there in the real world, there are bacteria called mutators, and they're pretty frequent. About in the region of 1% of wild bacteria where they have been measured, in the species where they have been measured, are mutators. They are bacteria with 100 to 1,000-fold increase in the spontaneous mutation rate. And they may actually contribute to a lot of the changes that you get in clinical isolates. One final thing, even if a resistance event is very rare, if it's out there somewhere, it can be selectively enriched once the drug is being used. Now, this is a messy diagram, but what it indicates is we have a combination of mutations, horizontal transfer, virulence factors, And all of these can come together in certain drugs, examples being ST131 and ST258 for E. coli and Klebsiella, which have become globally prevalent, highly drug-resistant organisms. And it's not just about the drug resistance, it's about the drug resistance in the context of something which spreads and is virulent and is difficult to get rid of. Okay, so I'm going to look at these 5 drugs or drug classes. The first 3 are classic single-target drugs, RIF, acetic acid, mesalamine, which Ursula mentioned, and then amino glycosides and fluoroquinolones, which in principle are multi-target drugs. And we look at what happened with those. Well, rifampicin, as I say, classic single-target drug, mutation rate pretty high, 10 to the minus 8, and that's because you have over 100 different mutations that can give resistance. Most of them give high-level resistance, most of them have a fitness cost, and resistance is expected to occur frequently during an infection, and it does. However, there's clinical evidence for selection against high fitness cost mutations, so fitness does matter clinically. There's also clinical evidence for genetic compensation occurring. Someone should take one of these buttons away. Okay, this is just a brief summary of the evidence. There's a lot of evidence, a huge amount of evidence now that in TB, where RIF is widely used, there's selection against high fitness cost mutations, but they do occur because they pick up secondary mutations that compensate the fitness cost. So these things are clinically relevant. And the context I want to put that in is you have a lot of bad things about rifampicin. A big mutational target size, high fold increases in MIC, a lot of fitness costs, but also a lot of compensation. And then you have to say, if you were inventing this drug today, would you continue with the development, seeing the high resistance frequency? Well, context here is that rifampicin has an incredibly useful clinical target product profile. It's among one of the most important antibiotics we have. But depending on how you look at it, it has a really lousy profile if this was a new drug coming out. Acetic acid, another classic single target drug, elongation factor G, also a high mutation rate, also associated with not just being single target, but also multiple mutations give the effect. Most of them have fitness costs. There's also clinical evidence for selection on the basis of fitness. There's also clinical evidence for compensatory evolution. In this case, you additionally have resistance associated with horizontal transfer, target protection. And the context here is that in the case of acetic acid, the high frequency resistance severely limits its use. It's primarily used for topical use. I know that it's currently being redeveloped for launching on the US market. We have to see, and it's been a moneymaker for small pharmaceutical company Lyon for decades in Europe, but it's limited very much in the areas where it can be used because of the high frequency of resistance. So this, I think, would be a classic case where if you discovered it today, I think you would think very seriously about whether you wanted to continue with this. But there was money to be made in this. Mesalamine, shouldn't dwell on it. Ursula's taken this up. Single target, PBP2. It's been used for decades in Europe. Resistance has consistently stayed quite low in the few percent level. And as she explained, there's a very high frequency of resistance, but you get very infrequent clinical resistance, and it's for a mixture of reasons. Non-expression of the resistance phenotype in certain clinically relevant environments or high fitness costs. And so, again, this is one, if you just based it on lab experiments, you might drop it, and you'd be dropping what's actually a very useful drug. Amino glycosides, these I think are really interesting because here you have the question, do we have a single or a multi-target drug? Well, in gram-negatives, which is probably where you would start looking if you discovered amino glycosides today, they almost all have multi-target. It's targeting ribosomal RNA, and for example, in E. coli, you have 7 different operons. That's the target, and you, to a first approximation, never ever get a resistance mutation in the target, in the ribosomal RNA. You do, however, get resistance mutations in a ribosomal protein, which is slightly off target but affects the target, and it's a single gene. And also, so you say, great, no problem, very few mutations, most of them are high fitness cost, you get it out on the market, and once you're out there, you discover there's an incredible number of drug-modifying and target-modifying enzymes out there in nature that become your problem. So your prediction of resistance based on your in vitro experiments would have misled you in this case. Doesn't mean it's not a valuable drug, however. But you take it into Mycobacterium tuberculosis, a different genetic situation. There's only one ribosomal RNA operon. And you do get mutations there. You do have selection for fitness. You do get genetic compensation. You also get the ribosomal protein mutations. Same again, you have selection for fitness. So fitness really seems to matter clinically, and genetic compensation does occur clinically. The big advantage in MTB is you have no horizontal transfer. So you have something where you have a predictable and relatively low mutation rate. You have a lot of mutants with fitness costs, and if you manage it carefully, you have a drug that I think has a lot of benefits. Clearly, it has a lot of benefits here, but the point I'm making here is that single versus multi-target isn't always obvious. You have to think of what bugs you're going to be targeting here, and depending on what you target, horizontal transfer may or may not become an issue. Final one, quinolones. The classic dual target drug, gyrase and topoisomerase 4, even if it wasn't appreciated when they were initially discovered. Low frequencies of resistance, but unfortunately, targeting is unbalanced. And depending on the organism, it's focused primarily on one or the other of these targets. And when you look clinically all over the world now, resistance frequencies to quinolones are, sky high. Some places they're right up at the top end. In other countries, they're 10, 15%, but it's unacceptably high in almost all cases. Why is that? It's not that you have a high frequency of resistance to a resistance mutation. It's that you get sequential resistance. And the example I've given here is from E. coli. You have multiple events that can be picked up with low selections. So selection with low levels of drug, weak selections. You pick up a first step, a second step, a third step. And then in the recent decades, we've discovered that there are also horizontal mechanisms out there in nature, which are now found in most of the clinical isolates, target protections and drug modifications and mobile efflux genes and so on. So here we have a multi-target drug that's effectively a single target drug. Resistance occurs typically in small steps. Again, how many steps depends on the species. And it involves, in reality now, horizontal transfer as well as fitness selection. I noticed you used this. I think Morten Sommer stole this slide from us because we had this published before him and we've been using it for years. But in any case, here we are, the same thing that Michael showed. You can plot resistance on a plot like this, where you look at changes in MIC along the bottom, relative fitness along the y-axis. And the space marked in orange there, the high-risk space, is an area where you have high fitness and high MIC, and if you have a large circle, you also have a high frequency. And this is, if you like, one of the take-home messages. You want to avoid this space. So given, as Ursula said, that most new drugs, for good or bad, are single-target drugs, what should you look out for? Well, you should look out for that high-risk space. You don't want a high frequency of resistance associated with a high fold change in MYC and associated with high fitness mutants. It's a bit harder to predict whether compensation would occur frequently, but if you have this combination, you probably want to be very wary. What can you do about it? Well, I think extensive MYC90 testing is probably advisable to see if you can uncover pre-existing resistance in the bugs you're targeting, because if it's out there, it'll inevitably be enriched. And this combination of MIC and fitness out there in the TPP organisms really gives you this risk for potential clonal expansion. You might also detect mobile elements within your organisms, but if they're not in the organisms you're targeting, again, as you mentioned, Morten Sommer saying, and I've thought of this idea as well, you could take every single piece of DNA on Earth and clone it in and ask, will it give resistance? You'd probably be able to identify, in principle, all possible resistance mechanisms, but we still wouldn't be able to predict the probability of them getting into your organism. So it would be a huge waste of money and resources. Multi-step frequency of resistance. Michael mentioned this. I think this is something very useful to do. Very often you just go in and you say, do I get any mutants at 4 times, 8 times, 16 times MIC? If the answer is no, you're happy, you tick a box. But we all know that in nature, in clinical use, bacteria will be exposed to low concentrations, and they will be exposed to different concentrations repeatedly. And so it's really important to know whether when you expose them to 1 or 2 times the MIC, you get some weak mutants, and then you re-expose them again. Do you get them to come up relatively quickly, or is it really impossible? And finally, if you run into a lot of problems, this is why I gave some of the examples earlier with TB and so on, you may want to ask, well, do I just throw away this compound, or do I start thinking about, maybe it has a use against a different organism? Maybe TB, maybe Neisseria gonorrhea, maybe Clostridium difficile. It's a different issue, but there are specific organisms where there's a lot of interest out there. And ultimately, for all of you in the business, it's going to be, a go, no-go decision. Do I take the risk now? There will be resistance issues out there. I think we accept that, but do I have enough information to decide I want to go forward, or do I just kill this project, leave it to somebody else? So.
The driver for resistance can be spectacularly different from the driver for efficacy in terms of reducing the burden. So you can have a situation, because you're looking always, when you do PK/PD experiment, for 1 or 2 log kill, you can have a very good driver because you're looking at 2 log kill, and yet because you don't necessarily look for it, you have actually enhanced or selected already resistant mutant in the thigh of these animals. And if you don't look for it, you don't know it. And yet, usually you don't cure a patient in 1 day. The PK/PD is always done in 24 hours. So you might, in that case, be encounter an issue. So you can have I can see a situation where you can have a bug where you see the 2 log kill and you're very happy, and yet you don't know, but you've selected also resistance.
I would like to add, this is a very common problem in old antibiotics, where we know exactly that the drivers are different. And one of the most prominent examples is continuous infusion of beta-lactam antibiotics, where we know that that may really favor resistance, but for a certain patient populations, it's much better to stabilize PK and also driving efficacy. So this is a common clinical problem.
This is something we can actually bring up in our own program, or when we talk to a customer or a partner. We can actually more and more, taking into account the role of the microbiome, we can offer the possibility of testing the activity of your compound on the microbiome. 2 reasons for that. One reason is that if you don't kill the microbiome and you kill only the targeted TPP pathogen, it stands to reason that your patient will be less sick from taking the antibiotic, but also just what you allude to a little bit, that maybe you will not exert selective pressure on the microbiome and favor horizontal gene transfer. That being said, I've not seen anybody really tackling that question directly. So asking the question, is there in the microbiome of humans already existing a gene that would confer resistance to your compound. I've not seen people actually facing that question.
So I think besides the human microbiome, you'd really want to broaden that issue to environmental release of antibiotics as well. Because I think the jury's out a little bit, but pretty much convinced that environmental contamination by antibiotics is a major contributor to the horizontal transfers that we see. So I think there are two different issues. One is antibiotic contamination driving horizontal transfer. The other, the microbiome issue, may be more focused on medical consequences, where we're still not fully aware of, the long-term consequences of killing off large parts of our biome on a more or less regular basis. But again, when you come into this area, there's, it's, in principle, I think you can make a strong argument for very focused drugs, but you come up against the economic issues then. And we already face, as far as I understand it, big economic issues with trying to develop broad-spectrum antibiotics, never mind very narrowly focused antibiotics. When you have a new compound and you do an MIC90 study, and you have, like Michael showed on one of his slides, a little peak off to the right or left, depending on which way we're going there, you have a real issue there. You know there's something out there that's affecting your drug, but you have a new experimental drug, and you may or may not know what the target is. Even if you do know the target, you can have a lot of difficulty trying to identify the cause there, because you're looking at clinical isolates, and you can put together a collection of ones with that same phenotype, and you really have very little to do except start doing a lot of omics work and hope that you get lucky and that you see a pattern that helps explain it for you. And I think it's very valuable, but there's no definitive way of knowing that you will get the right answer.
And just to echo on that, I agree with you. The history of the strains could be the genomes, but then it comes to the number of strains that you need to have in order to make sense of the data. And then we're talking about genome-wide association studies. So, and GWAS, and that's why I was referring to Alex, because it's very, very complex, and it's going to be very compound specific. So I was alluding to a case where with only a few genomes, we had a lead, an idea on what in those different clinical strains, there were all cystic fibrosis strains, and so very severely mutated, and we could, just by sequencing 20 strains, we could have an idea, but we've tried that in other cases, and we sequenced 140 strains, and you've got 140 genomes, 70 with low MIC, 70 with high MIC. You look at that and you're like, no idea.
But it may still have clinical implications, because these are exactly the cases where we need a new drug. So it should not be neglected. No,
you're absolutely right. And sometimes, actually, in that case I'm just referring to, there was an association that was very significant statistically with body site origin. So you're like, again, we don't know why, but and we also used biolog experiments, and so you could correlate also the ability to grow in one condition for some of these clinical strains, and so you've got correlation that are by no means causation, and we don't know what they mean. But actually, the clinical information that you alluded to sometimes can be correlated, and then it's interesting, and the phenotypic also can be correlated more easily than genome.
It must make a huge difference whether you're very early stage or middle to late stage, for a very trivial reason. The experience I've had from working together with companies because I'm in the Enable Consortium, working together with companies, is that at very early stage, compound is extremely limiting. And already at that stage, you're making decisions on whether or not to pursue a certain path or not. At later stages, yes, you can do incredibly extensive MIC90 panels and any studies you like to try to figure out where you want to end up. But at the early stage, I think you have a real problem with quantities, because the chemistry may be difficult, it may be expensive, and you're going to do the simplest, least expensive assays possible, and you're going to end up making decisions based on those. Now, I mean, I think that probably means there are some poor decisions have been made in the past. There may be compounds that have been discarded because they were checked on one strain, they gave a high frequency of resistance, that's the end of it. And so I don't know what the experience of small companies here is. How easy is it to feel confident that you're making the decisions correctly? And maybe to Michael here, because you're in larger pharma, how possible is it to have an encyclopedia of all of the compounds that have been checked in this field before, if people want to go back to them?
So having the encyclopedia, as you say, it's difficult because we routinely, because we are deeply involved in natural products, we routinely look into very, very old papers where some of, as you said, some of the compounds have been stopped because of high frequency of resistance. We have obviously there is no data bank that I know of, at least, where all the MIC and the frequency of resistance are put next to one another. And besides, there is no harmonized protocol for frequency of resistance. For MIC, at least we have CLSI and UCAST guidelines, but for frequency of resistance, no protocol. So I think it will be extremely hard to have what... It's interesting, but very hard.
I mean, I think the context here was illustrated by... Is this on? That's all, yeah. Illustrated by the thing like mesalamine, for example. You know, if you had just discovered this drug and you got this 10 to the minus 5 frequency of resistance, you would just be scared and drop it. And would you go to the trouble of purifying or making more of it to do more extensive studies? I think this is where the issue is, especially as we're trying to discover completely new compounds and new targets. What's the risk that we get scared off far too early? Usually, if I'm talking about fitness, I'm very careful to say relative fitness, and relative to a comparator, but also relative to the environment you're testing it in. And I think this is where you have the issue. But in principle, you have the same issue with MIC. I mean, what's the relevance of MIC? It's a number measured under pretty artificial conditions, but everybody agrees to do it that way. And to a large extent, it is predictive of what is likely to happen in vivo. Not completely, but I mean, we continue using it because we can extrapolate from those values in most cases. And so maybe if we're worried about fitness, people say, oh, what's the relevance of measuring fitness in LB as growth rate? You know, this is totally irrelevant. But I think all we need to do is to build up a database where we can extrapolate from that. And the examples I gave there, the ones from, say, aminoglycosides and TB, from fusidic acid, from, I can't remember what the other one, the quinolones and so on, they do actually correlate well between what you see as frequencies in clinical samples and what you see in simple in vitro lab experiments. And so I think we could work together on this. This is the kind of thing ECMID would be good at. You could have a working group trying to gather data and establish agreed protocols. And there will always be exceptions, but in principle, it's not worse than MIC.
Okay, so I can tell you what we do. Okay, but that's by no means a guideline. But there are several things we do or we can do. First, fitness, we do just growth curves. In MH2, you look at the growth curve and you see if your mutant, a recent mutant, has actually different growth curves compared to the parental wild type. Okay, and the longer it takes for the in vitro for the bacterium to grow, the less fit the bacterium is, of course. We do also TKC, so that's without pressure. We do also time kill curve experiment in a mixture of your mutant and your wild type at different ratio, 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, at different starting inoculum sizes, 10 to the 5, 10 to the 6, 7, and 8. And then we put MIC, time twice MIC, 4 times MIC, 8 times MIC, a lot of plates. But you do these time kill curve experiments, and then you see in which cases you've got a ratio of mutant to wild type that you cannot cover by your drug. We do in vivo fitness, of course, also in that case where you just take the bacterium, the mutant alone, and you see if whether it's establish an LD50 of that bacteria alone. Then we do also competitive indices, so mixture of wild type and mutant 1-to-1. Grow them in the animal, and then you pull out and you see if there is a displacement of the mutant versus the wild type. Without treatment, with treatment. So that's for the in vitro and in vivo fitness without pressure and with pressure. The last thing we do also is to use to establish the growth curve of the mutant in a wide variety of growth conditions. So using the biolog machine. So sometimes we've got surprises that your mutant is actually less fit in MH2, but much more fit in at low pH for whatever reason, or with some sugars, it's actually more fit than the wild type. So you're like, wow. So lots of data. What do you do with that? Frankly, we use that because as was said, I was in big pharma, so maybe more means, but we were profiling several compounds, and then you just choose the better one. That's the only thing. It doesn't really tell you whether you've covered your basis, but at least you make all chances. You put all the chances on your side.
Simply testing it in urine if you're targeting.
Sure. So we routinely test so what we call the vivo mimetic media. So we have urine, serum, carbonated MH2, iron-deprived medium, thing like that.
Are there more questions? Maybe just a comment from my side. You mentioned the concentration in the clinical situation and PK/PD and maybe compensating with dosing. So then the new compounds that I see have, in general, rather high MICs. So very common is an MIC 90 of 2 to 4. That would mean in PK/PD questions that you need really very great pharmacokinetic characteristics to have a reasonable PK/PD study. And this means just efficacy. If you're going to the mutant prevention concentration, that's usually much higher. So this is the big question, if that's possible with pharmacokinetics, and this correlated, of course, to toxicity. So you need to do that. To be able to do that, you need great PK, and you need great safety. And I'm not sure if you can assume that with new drugs. So speculating that you can make up with high dosing is maybe not very realistic in most cases. Are there any more questions? If not, then I think we are really good in time. Then I would like to say thank you, also on behalf of my colleagues here. If you have more questions related to that, then I'm sure we'll find a way, maybe on the GARDP, on the Revive website or something. And we will also make sure that you get the presentations, and so, you can benefit from that also at home and think about more questions if you have more. So enjoy the conference, and thank you very much for coming.